Okay, and welcome to the single cell biology session. I'm Marg Sutherland. I'm the program manager for the Neurodegeneration Challenge Network at CZI. And I have to say, a lot of our investigators are doing single cell biology and using some of the software you're going to be talking about today. So we're excited to hear more. And with that intro, Paul, do you want to take it away in terms of telling us about Azimuth?
Yeah. Hi, everyone. My name is Paul Hoffman. I'm a bioinformatician with Rahul Satij at the New York Genome Center. I'm a developer of the Seurat R package, and today I'll be talking to you all about a new tool we recently released called Azimuth. So what is Azimuth? Azimuth is a Shiny R web app built to map single-cell RNA-seq datasets to a reference dataset. This is the first tool that our lab has built to enable single-cell analyses without any knowledge of the R programming language, so we're rather excited about this. Using Azimuth, you can go from a raw data set to annotated clusters without using any R, and this whole process is a lot faster than the traditional single-cell analysis workflow of reading the data in, normalizing, running dimensional reduction, supervised clustering, and manual annotation. Most data sets can be mapped and annotated in just a few minutes, and Azimuth is also able to impute the expression of alternate modalities present in the reference, such as protein expression. In addition, we also provide Azimuth as a service funded by CZI. We've set up an Azimuth website where we're hosting a reference data set that we've generated of around 210,000 single PBMCs that can be used for mapping a query data set. And we allow users to upload a data set of up to 50,000 cells to this website, where we'll perform the mapping on this infrastructure. After a few minutes, users are able to then download the cluster annotations, scores to see how well their data set mapped, as well as the imputed protein expression that Azimuth is able to generate. Just as an aside, this data set, this limit of 50,000 cells for upload is a limit that we're imposing on this web service, Azimuth itself, and the mapping algorithm that backs it is capable of handling much larger data sets. And so now we're going to go to a live demo because these always work well. So here I just want to walk through some of how Azimuth works and what you can do with it. So I've already uploaded a data set of 3,000 PBMCs, which is generated by 10x Genomics and is often referred to as PBMC3K. If you don't have a data set that you want to upload, you can load a demo data set that we provide, which is 11,000 PBMCs, and use that for mapping instead. So I'm just going to start the mapping real quickly, which is a one-button process. Azimuth can take a wide variety of input formats. We support the 10x Genomics HDF5 format, as well as a matrix or data frame of count matrices stored as an RDS file that you generated in R. If you have additional metadata for your data set, such as a donor ID, disease status, or cluster labels from a previous analysis, you can upload your data stored as a Seurat object, either in an RDS file or an H5 Seurat. Or for Python users, we also support AND data objects stored in H5AD files that were generated with AND data 0.7 or newer. In terms of QC, we allow users to filter based on the number of molecules or N count, the number of features expressed or N feature, as well as the percentage of mitochondrial genes that are being expressed. In these QC violin plots, the red boxes will actually live update as you adjust the filtering to be more or less stringent. Once the mapping completes, which this data set did in 34 seconds, we can actually start going through the results. So Azimuth will provide a pair of dim plots for the reference and the query. Both the reference and query are projected into the same UMAP space to keep the visualization consistent between the two data sets. And if you upload a data set with additional metadata, you can actually switch the query coloring to that other metadata so you can see how your previous results fall in the UMAP space and compare to the reference data set and the labels there. In addition, we also provide this contingency table where you can look at the percentage or frequency of any two metadata columns, such as the predicted ID and a previous clustering result. In addition, we can also explore the data set, the query data set on a feature level. So here we have both a feature plot and a violin plot for a gene present in our data set, in this case GNLY. We can actually adjust this to any gene that we want. So here's CD79A, and it will update both plots. We can also choose an imputed protein. The query data set doesn't have any protein data, but we're able to impute protein expression based on from the reference onto the query. And we're also able to view the predicted ID scores. This one is how well the data set mapped overall, or we can see how well a specific cluster from the reference data set, how a specific cluster from the reference data set maps back to the query. In addition, we also have these two biomarkers tables, both on the RNA that was provided in the query, as well as the imputed protein biomarkers. Differential expression is done using the Presto package, and you're able to click on any one of these rows and have them automatically update on the plots above. Lastly, we offer several downloads from Azimuth. So you can get a table of the predicted cell types and the prediction scores, both the whole mapping wide score as well as the per cluster score. We provide the UMAP coordinates in a Seurat dimensional reduction as an RDS file, and we also provide the imputed protein expression as a Seurat assay. And all of these you can easily load back into R and store in a Seurat object for further downstream analysis. We also provide a template analysis script that'll walk through how to do the analysis in Seurat, and it will automatically filter, it'll automatically pre-populate with the values that you put in for the filtering. And somehow get back to slides. So where do we want to go with Azimuth? Well, in the near term, we're looking at building additional references for more tissue types. Currently, we only have a reference for PBMC, and so it really only works for these types of cells. But we'd like to build it for more tissues so that way we can support a greater variety of data sets. And as we build these references, we plan on deploying new instances of Azimuth for the community to use for each of these different references that we built. In addition, we're also working on the ability to transfer multiple levels of annotation, inspired by the way the Allen Institute provides annotations for their data sets. We'd like to be able to transfer sort of multiple resolutions of cluster labels to different data sets, to data sets that are uploaded to Azimuth. In the longer term, we'd like to offer support for self-hosted Azimuth deployments. Right now, the stack to deploy Azimuth on a server is rather complicated and it's not very automatable. But our lab itself has benefited from self-hosting services such as RStudio Server, Jupyter Hub, and UCSC's Genome Browser. So it'd be nice for us to be able to give back to the community and provide a self-hosted Azimuth so that way people could easily deploy it and use their own references without relying on us to build that for them. But this is not something we have much experience in. So if you guys know how to set up easy deployment, easy and automatable deployments, or you know someone who would be interested in this, we'd be happy to talk to you. This work was done in collaboration with several members of the lab. Yuhan, Andrew, and Charlotte in particular helped a lot with building the mapping algorithm and getting the app built and deployed. If you'd like to play around with our reference data set, the full reference is hosted at the Fred Hutch. You can view the source code for Azimuth on GitHub. All of it's publicly available. And if you'd like to play with the app itself, you can go to our website /Azimuth and either upload your own data set or use the one that we provide. Thanks.
I have to say, one thing you already asked or answered one of my questions, and that was when you were talking about the PBMC reference data set. Our investigators are into doing single cell in brain tissue in that. So I was wondering how the reference data set for the PBMCs would interact with a data set like that. So
the different tissues tend to be harder to map, so that's why we're looking at building new references.
mm-hmm.
Looks like Robert has a question.
Yeah, Robert.
I was wondering to which extent would it be possible to, oh man, my question is full of typos. Anyways, it's because the keyboard is far away from my position. To which extent is it possible to deploy Azimuth already containing a couple of data sets so that you can have multiple users analyze or take a look at those data sets without having them re-upload their own data set every time?
Where the data set's already provided?
Yes, so let's say that somebody wants to provide a couple of data sets and allow other people to analyze them, but without having them to go through the hassle of uploading it. Is that currently possible?
Azimuth currently allows just a single demo data set. We don't really have support for multiple, but that would be an interesting thing to build in. That should be doable, and that way you would then be able to deploy an instance of Azimuth with multiple data sets rather than just one.
Yeah, the reason I'm wondering about this because it sounds, I mean, having the tools to be able to go through a single cell analysis without any programming experience is especially useful for people who can't do this, but then it would also be useful to have some kind of service platform of people who analyze the data sets or process the data sets and all put it into Azimuth, whereas then the non-tech savvy users can then analyze it without any hassle.
That is not something we had thought about, but that sounds like a good way to go. Thank you for that.
All right, cool. It sounds nice that you're thinking about it.
Paul, so you mentioned you talked a little bit about the cap on 50K and how that's just sort of a limitation of the current web instance and not an underlying limitation of the mapping algorithm. Can you talk a little bit about like scalability as the size of the reference data set increases? Like if we have 10 million cells from everything, is it going to take a lot longer or not much longer, or that kind of thing?
That's a good question. I don't think we've really explored the effect of mapping or the effect of reference data set size on mapping. I don't think it should take that much longer because most of the work on the reference side is actually pre-computed. And so we're just pulling a model basically from the object and using that in the mapping algorithm. So if I believe what's happening is that it's more dependent on the query size, and the time to generate the reference does depend on the size of the reference, but once you have the reference generated, that shouldn't affect any downstream mappings.
Awesome. That's good news because, there's a lot of data out there and more all the time. Yep. Cool. Thank you.
I was wondering, for the mapping, do you use some kind of cell ontology to structure the cell annotations for the mapping that's being generated?
So the labels that get transferred are just transferred directly from the reference onto the query. And this reference that we built, we worked with a lot of experts at NYU and the Fred Hutch and other institutions in order to figure out what cell types are these actually, what's the best possible label for these. And so it's been a couple years in the making, this reference data set, just going back and forth between different collaborators and asking for their input as to what these labels are. And that's just for this one. If you were to build your own reference, then it'd be the labels that you would decide are best for those cells that would get then transferred onto the query.
Okay. So it would be possible to use your own, use an ontology where the cells are being described in.
Mm-hmm.
The nice thing about using an ontology rather than fixed labels is that perhaps then you could allow the user to choose the granularity in which the different cell types are being displayed,
Yes.
rather than having. Okay, yeah.
That's one of the things that we are working on, and that should be live in Azimuth, at least technically live in Azimuth, but not for this reference per se, within the next month or two.
Amazing. Okay, thanks.
Mm-hmm.
So we are going to talk about Scanpy. So Sega and I work as so I'm a postdoc in the Institute of Computational Biology, located in Munich, Germany. So it's late at night here. And first of all, as promised, I wanted to tell you about the shrimp in our logo. Think of a different word of shrimp. It's probably Scanpy. And then you can make the wordplay of Scanpy, Scanpy. Yes, I know it's ridiculous, but it's funny too. Okay, this is where the shrimp comes from. So Scanpy is the single cell analysis platform in Python. So we work entirely in Python, and it was predominantly originally developed for transcriptomic analysis. When we think of single cell transcriptomic analysis, RNA sequencing data, we have thousands of cells and thousands of features, and we have to walk through lots of steps in a pipeline to make sense out of the data and recover patterns. So luckily you can do all of these steps in Scanpy. So going from raw preprocessing, raw preprocessing is done with cell ranger usually. So we focus on, UMI data. So we have the count matrices, you can do the quality control, you can do anomalization, you can do batch correction, select highly variable genes as interesting features. You can visualize your data in various embeddings, then use a clustering algorithm to find interesting groups of cells, identify marker genes, or use marker genes, then to annotate the cluster, make sense of the data, and then finally go into several downstream questions depending on the data set that you have at hand. So you can, for example, do differential expression tests on cells from different conditions and see what's differentially expressed, but you can also do compositional analysis. So imagine the main driver of disease is actually a change in the cell type abundance, and this is then part of this compositional analysis. Furthermore, you can go into trajectory inference, so meaning that you can follow the maturation of immature cells towards a mature cell. And here we have a couple of algorithms developed. One is the PAGA algorithm, where you see how clusters are connected, and then you can move along a certain trajectory from in your data and to uncover gene dynamics or metastable states. There's a lot of algorithms out there for the analysis of transcriptomic data. So at some point, we also had the feeling to be a bit lost with the variety of algorithms. So we wrote this current best practices in single cell RNA sequencing analysis that comes with a tutorial, an extensive tutorial, I have to say, because Malte is, so the first author, is a very meticulous person. And you find every detail you ever want to know about single cell analysis in this tutorial. It's written in, it's written in Scanpy, of course. All right, so let me quickly show you a couple of recent additions, and we had a major overhaul on our visualizations. So for example, if you have preprocessed your data and now you want to plot a UMAP, you can go into this with SC stands for Scanpy.pl stands for plotting, and then we go for the UMAP. And what comes up is this very, what I find pretty annotation of a couple of the data set. And now if we want to annotate plot marker genes, for example, we go with that function, say scanpy.pl.plot, we give it our data set in the end data format, give it a marker gene dictionary, and tell it which groups to plot. And what comes out is this amazingly pretty dot plot that you see here, where the dots have the size of the dot represents the fraction of cells that actually express the gene, and the color shows you the mean expression in each of these groups. If you're not really keen on dot plots, you can go for the stack violin plots, where you have essentially the same information, but you see not just a dot, but the actual distribution of the gene expression in each of these clusters for each of these genes that you want to visualize. The more classic view that we had until recently, ever since, is this matrix plot, where you see the mean expression for each group. So it tells you a bit less of the information, but it's really nicely organized nonetheless. And what you also see is that you get really complex plots with a very simple syntax. So that's the small plots. I also just quickly want to show you, I would say, a really, really nice heat map. We also have tracks plots. And all you have to do is just call the strength genes groups heat map plot, give it a couple more parameters to finally create this. But you can, so what's the basis of these plots is matplotlib, so you basically have the full control of what and how you want to visualize your data. If you want to see more of that, we also have tutorials on the various topics, including the plotting. So where is this going, Scanpy? Scanpy or NData, the data package that Scanpy is operating on, is already used in a variety of other projects, like the CellXGene browser. It uses the H5AD files to display an interactive map of your data that you can explore without knowing any code or any coding. SCVI tools uses Scanpy as a basis, and also the Human Cell Atlas provides its gene cell browser in the Scanpy format. We also worked on interoperability of Scanpy. So we recently, in a project together with Robert Canot, who's also presenting today, we have enhanced and simplified the integration of Scanpy in R with the NData R package. If you want to load your Scanpy package, or not the Scanpy package, but the NData files that you did in Scanpy, and you want to try out some functions in Seurat, there's this free H5AD function that you can simply load your data object in Seurat, and vice versa. We provide the NData to our I package to convert Seurat objects in the Scanpy format. We're not only developing Scanpy, but we also allow and encourage others to develop towards the Scanpy ecosystem. So here I named two functions for data integration that work in the Scanpy ecosystem. So one is BVKNN and the other is Scanorama, where we recently enhanced the integration for. Then when you want to work on gene dynamics and work with RNA velocities, you can go for the SCVelo or the cell rank package for the cell fate mapping. Finally, we also have perturbation analysis with SCGen, and we also provide label transfer similar to Azimuth with the SCArches project. Scanpy doesn't do not only transcriptome, but also epigenetic marks with the EpiScanpy package that is developed also at the Institute of Computational Biology by Anna Denese as the lead author. And Scanpy goes spatial. And what this is about will Sergey tell you more in a minute. If you got curious and want to try out more for Scanpy, here are a couple of resources you can play around with. So we have the Scanpy tutorials. on the Read the Docs and our best practices workflow on our GitHub page. If you want to stay tuned on our news, please visit the Scanpy Read Docs webpage or follow us on Twitter. We are also going to set up a Slack channel to build up the Scanpy community for more contribution. And with this, I would like to hand over to Sergey.
Yeah, hello to everybody. I'm Sergey, one of the developers of Scanpy. And today I want to tell you about some developments around Scanpy. First of all, I want to advertise SquidPy. It's a package for analyzing spatial transcriptomics data, which will be released soon, like in a few weeks. It's fully integrated with Scanpy and Undata and uses many of their capabilities for visualization and analysis. This package provides data loaders for spatial data sets, functions to build a spatial graph, test the statistical significance of neighborhoods, do segmentation on images. For example, in the bottom left corner, you can see how the package allows you to identify cells in spatial spots. And this package provides many more functions. So I hope you're interested and will try it and use, then it will be released. Next, I want to move on Undata, what is happening now, what was implemented, and what is in process. A few words about Undata objects. Undata is used by Scanpy as a data container, and I mean, it's a separate package, but it's fully integrated with Scanpy, and it's also used by other packages like SCVI, for example. You can see a visual representation of an Undata object on the slide. We have a set of observations for which some of variables have been recorded, and we want to build an understanding of this data set through analysis. So we add annotations onto our observations and variables through annotation with outside resources and computed representations. The data set plus these annotations they are collected within an Undata object. As a backend, we use this hierarchical array stores, which are HDF5 and ZAR. They are hierarchical data formats. Then they use a file directory-like structure that allows you to organize heterogeneous and complex data in a structured way. We mostly use HDF5 as a storage container for Undata objects. These are H5AD files, which I think you know. But we also support ZAR. These hierarchical array storages, they provide you fast partial access to your data and compression. So I want to tell you about how we represent our Undata objects on disk. So this you can you can see, for example, with H5 ID that every object in Undata class stored differently, which simplifies partial reading of these objects. We strive to provide easier partial regions of objects and out-of-core computation, and this is a work in progress. Also, many people which work on different packages using Undata were asking us about storage of custom objects in H5ID files. And now we are working on providing this possibility. On the slide, you can see an example of how you can add add a saving capability, saving and reading capability for custom objects to data. Basically, you register functions for writing and reading this data, and then after registering, then data is able to save an data object with these custom objects to a file. I mean, like a more complicated example, but I will not stop at this. Another thing is, for example, the thing which I'm working on now is a shared indexer for data and a PyTorch data loader. By shared indexing, I mean that then you have several data files on a disk, for example, and you want to work with all of them at the same time. But for example, there are too many files, and you want to avoid the loading them in memory and concatenate it. You can use this functionality, which is in progress, for basically providing these addresses of these files. It will load them in an dataset object and will provide you the ability to basically select, subselect all the objects at the same time and get the underlying annotations and data without copying the whole things in memory. And the second thing is that you can, for example, pass this object to a data loader, and it will work conveniently with data loaders for PyTorch, for example. Yeah. Thank you. I think that's all from my side. If you have any questions, please ask them.
Very cool. Thank you. And this is, at least to start with, Morin, a great idea about having a tutorial that new users could tap into in terms of best practices. We're working with our network to introduce them to some of the software packages and that. So that's a great idea.
I was really interested to see about the custom data types. Is that something that's already in Scanpy, or is it currently in the development branch of on GitHub?
It's in development. I don't think it's on any branch, but it will release soon. It's a part of Undata.
And so when I want to use this, I need to write a Python package where we have some extra annotation about the data objects. And like, what happens if I try to put something else inside of that field or whatever?
Yeah, I think it will throw an error, but it depends on the object. For example, if it's something simple like NP array, or I mean, we support NP arrays and pandas data frames and so on. But if you have something like really custom, then yeah, you have to wait for this functionality to be implemented to use. But
sorry, continue.
Yeah, but I mean, then you have it. You can just register your object, write a function like how it saves it to a HDF5 group, and it will save it and read.
And then how do you access it once it's inside of an H5 object?
Just read it with an data read. Yeah, if you registered the function to read it, also it will just read it.
But like, is it stored inside UNS or
inside? Yes, inside. It's inside HDF5. It's in the group. In the group, yes. I mean, it depends on the object, but normally it's stored in the group. Yeah.
Okay. Well, let me tell you why I'm asking this, because I'm working a lot with trajectories, as you might know, and this kind of data objects. Well, it contains both information on the topology of your trajectory, as well as where cells map onto that trajectory, as well as some other stuff that can be of interest. So what I've been using on data for now, I kind of need to spread all of the information that I have regarding to my trajectory inside various groups, such as ONS and VAR and this and that.
Yeah, this makes sense.
But so this would mean that I can store a trajectory object inside of the OnData, and then
If it doesn't make sense to store things in the OBSM or VARM, yes, you need UNS. If you need UNS, for example, you can, for example, create a custom object in UNS, register a processor for this, and yeah, you will be able to use it basically.
Okay, I'll look into it a little bit more myself. I'm really interested.
Thanks. That was really interesting, and I'm interested in the AND data container. So, because I'm coming from the bioconductor world, so using the single cell experiment a lot, and one thing that I found to be nice when teaching is with the single cell experiment, you can with like one line index based on a location. So you can say like, give me the expression nearby this peak or something like that. Do you think you'd build out, and I'm sorry if I missed it, but could you build out like indexing based on location?
We have also this work in progress. It was postponed a bit, but finally we will have it like once. Yeah, but it's in progress. Yes, with path like access in and data with paths basically. Yeah, what you mean? Yeah.
Thanks. Looks great.
So I'm really interested in the efforts to move to out-of-core computation and just generally being able to work with very large data sets. Even with a data set that is not millions of cells, we're sort of only getting away with it because the matrices are so sparse. But if you want to do things like, variance stabilize your data or subtract some batch effect coefficient or whatever, now all of a sudden you're looking at the residuals with respect to some model and it's no longer sparse. So have you put any effort yet into kind of transforming very large matrices and then storing the output on disk to facilitate out-of-core computations later on? Is that working, or is there anything that is unexpectedly hard about that?
We have some out-of-core capabilities with Undata now. You can read Undata in a bucket mode, and it will lower everything except X, basically your main data matrix, which will be on disk. You can still access it, like selection, and also do some pre-processing and plotting. But I think Scanpy doesn't provide the full functionality for this type of computation now. But we are working on it, and it's also in process, and it will be also provided, I think. But for now, we have partial functionality for out-of-core computation. Yes.
Cool, thanks. So I'm Cole. I'm from Seattle, and I'm going to tell you today about Monocle. But I looked at where I was in the lineup, and so I figured I would probably not need to do a lot of introduction on the types of analyses that folks do with single cell genomics, because there is a lot of overlap between Monocle and both Scanpy and Seurat. I would say that Monocle is it's one of the oldest dedicated packages for single cell genomics analysis. Old being like 5 years. and historically, the focus has been on trajectory analysis for developmental biology applications where you can kind of resolve fate decisions and things like that are happening. But we also do clustering and differential expression. And Monocle has been around for a while, but it's been wholly rewritten at least 3 times. So it looks very different than it was, and much better than it was when I wrote it as a postdoc a couple of years ago. So I'm not really going to talk about the tasks. What I want to focus on today is how we are trying to keep up with the ever-increasing pace of technology development on the experimental side. Because one of the reasons we had to write Monocle 3 times was because single-cell genomics technology has changed. And in particular, it's changed in terms of scale in a really dramatic way. So if you're into this field, you've probably seen this figure from a perspective written by Valentin Svensson a couple years ago, where you can see that just by sort of different ways of doing single cell genomics, we have, as a community, gone from being able to look at a few hundred cells in a given experiment to looking at hundreds of thousands of cells or even millions of cells in a single experiment. And my lab, actually, I would say, even though we started out as kind of all dry, something like 75% of my lab is now actually involved in experimental tech dev. So we, together with Jay Shinduri's lab, have been heavily focused on this latter category called in situ barcoding over the last couple of years. And just as an example of the scale that this way of doing single cell analysis, what you can get with this, using Sky RNA-seq, we were able to look at the transcriptomes of more than 4 million cells from each of 15 different fetal tissues, human fetal tissues. This paper just came out like 3 weeks ago, and it was partly supported by CZI, so thank you, CZI. And so this is a huge data set by single cell genomic standards. It's millions of cells. I think that what's coming down the pike is going to be, experimentally are even more dramatic changes. So I think that many labs will be able to routinely do experiments with millions of cells, and probably from hundreds or even thousands of different specimens. I think that increasingly we'll be using single cell analysis to analyze complex systems like organoid systems or whole organs or even whole animals. Our experiments are going to have complex designs where we're perturbing the systems in different ways or looking at lots of different genetic backgrounds. You've already seen a little bit of preview of this. I think Paul alluded to this for sure, that we're going to be able to read out multiple different types of data at the same time. So not just gene expression, but gene expression plus chromatin plus protein. We've seen in the past 2 talks that there's this increasing move to spatially resolved experiments. So all of these changes require big changes to the software stacks that we use to analyze these types of data. So I just want to go over our plan for keeping up with the times in Monocle. The first thing that we're doing is just improving interoperability between Seurat, between Monocle and Scanpy, taking advantage of the work that the dev teams for those tools are doing, other tools as well. And just as an example of how this is already going on, in a parallel paper to the one that I just mentioned, we looked at almost 1 million cells worth of chromatin accessibility data from the same fetal tissues. And in this paper, we used Monocle, but we also used Seurat very heavily. And we were able to go back and forth between the two, in part because of the work that the Seurat team has already done in making their data objects sort of loadable and castable to Monocle objects and things like that. So thank you very much to the Seurat team. So we have plans to add sort of functions for reading and writing between different tools, sort of native containers, but also for the kind of storage formats like you just heard about from Sergey. So that's definitely on our agenda. The other is an effort to increase the accessibility of analyses that are done with Monocle. And our idea for this is to publish these kind of what we call study microsites, actually a little bit similar to what Paul talked about with Azimuth. So as part of the human fetal work, we posted this portal called Descartes, which makes the RNA data and the chromatin data accessible in an easy way so you can go and find your favorite cell types and tissues and so on. And you can go to this website right now and check out the data. You can poke around, you can download everything, you can download lots of the raw data and analyses. And you can even get into sort of assay-specific views like analyses that are unique to the ATAC-seq and so on. And all of this web infrastructure was built by Michael Zager, who's a collaborator of ours he directs the Fred Hutch Data Visualization Center. And he's built this framework that allows you to essentially stand up a website that looks like this, given a bunch of analysis files in the right format. So one of the things that we're going to be doing to Monocle is adding functions to serialize analysis objects in those formats so that we can push them essentially to Michael's sort of cloud-based infrastructure, generate a microsite, and publish it to the web. Interpreting these huge experiments is a real challenge, and one of the ways that we see as being potentially very useful is to move from a completely unsupervised analysis to a kind of a more supervised analysis. So as an example of what I mean, we did this drug screen last year and read out what happens when you perturb individual cells with lots of different compounds. And one of the things that we saw was that some drugs affect individual cells that are otherwise identical, same cell line, same culture well, differently. So for example, apothelone A, which is a microtubule stabilizer, when you treat these lung cancer cells with this drug, you basically get one of 3 different outcomes. And that's pretty cool. And it probably has to do with the mechanism of the drug and how it messes with cytokinesis. But one thing that you should know about this is that, one of the most important signals just explaining variation in the single cell data is the cell cycle. And, probably what's happening here is that how a cell responds depends on where it was in the cell cycle. So it would be cool if you could kind of subtract the cell cycle, the signature of it, from these data. And if you did that, you might imagine that these 3 states might collapse to a smaller number of states. So, we want to be able to kind of interpret our variation across single cells in terms of known a priori biological pathways and things like that. And there's lots of literature on how to do this kind of thing. There's these latent space models that project data into latent spaces that are defined by known pathways and gene sets, like PRISM is an example of that, but there are others. So we're going to be experimenting with different ways to do this in a super scalable fashion over the next year. I mentioned really large scale perturbation experiments, and I just gave you a little bit of a taste of one just now. But there's been some big, I think, advances in what are called cell hashing technologies that enable really huge experiments. So we have one, Rahul's worked on one with Peter Smybert from the New York Genome Center. Actually, they introduced this idea, I think. And the idea is just that you have cells from a bunch of different specimens or cells that you've perturbed in different ways. You label those cells with something that will show up in your RNA-seq libraries, and that allows you to pool the cells from all the different samples together and sequence them in one pot and then demultiplex the experiment after the fact. So we did an experiment like this with our technique called Skyplex, where we were able to look at 3 different cancer cell lines, each of which was treated with one of 188 different compounds over a range of doses, and we did the whole thing in duplicate so that we don't get horrifying batch effects and things like that. So that's like 4,500 different culture wells, and we were able to sequence about 500,000 cells from this. And so in each one of these kind of perturbations, we're like fitting a little regression model that is designed to see whether there are dose-dependent effects on each gene and so on and so forth. So we're doing all this statistical analysis, and now we're looking at even more complicated settings. Instead of looking at cultured cells, we're actually looking at whole zebrafish embryos. And in this experiment, we've generated a kind of a time series of zebrafish development at multiple temperatures, because temperature affects the the rate of development and progression. And through hashing, we can actually label each cell by which embryo it came from. So this is wild because it means you can kind of control for embryo level effects. And you get these incredibly complicated UMAPs and things like that with all kinds of different cell types. But when you want to sort of analyze the gene expression dynamics across development or in response to perturbations, you're going to make little regression models and things like that. And in an experiment like this, you might not want to model all of the different variables as having fixed effects. So one thing that statisticians often do in these sorts of scenarios is use mixed models where some of the variables are modeled as random effects. The cost of doing this is that it's slower than a sort of a simple regression approach. And so anything that's slow is going to be problematic when you have 1 million cells from 1,000 different specimens. So this is another thing that we're going to be focusing on in Monocle, is enhancing the regression infrastructure so that we can use more complex and sophisticated models, but still use them on really huge data sets. And then finally, I mentioned spatially resolved data. We have a new technology coming out from the lab that the paper's not out yet, but hopefully it will be soon, which basically is a way of doing single cell RNA-seq or single nucleus RNA-seq, but where you get spatial information about where each cell came from in a tissue. And the way it works is that we have these glass slides, and we've printed onto them a grid of labeling oligos, just like we use in Skyplex, so that each spot has a unique label. And then we just kind of sandwich one of these slides together with another slide that has a thin layer of tissue on it, like from an embryo, like a section. And then what will happen is that the labels will just kind of diffuse off the glass slide and onto the tissue slide. And then when we pull the nuclei out of the tissue, we can look at the oligos and the labeling oligos and map each cell back to kind of where it was in the tissue section. So we've tested this out in mouse embryos. And when you first look at these data, they look just like regular single cell RNA-seq data, and you get a bunch of cell types and all that. But the advantage of this is that almost for free, really, in terms of the cost of the experiment, you can also tell where each cell was in the section that it came from. So you can see that the cardiomyocytes, came from the heart, the neurons come from the brain and the spinal cord, the hepatocytes come from the liver, and so on and so forth. In addition to getting these kind of virtual in situs like we've come to expect from lots of different spatial transcriptomic technologies, you can look at this gene, for example. This is a dopamine transporter, so it's really only in this one spot in the brain. It looks very similar to what you'd get with a conventional in situ, but this is a single cell assay, and it's resolved at cell type resolution. So we can tell how much each cell type contributes to each gene's expression at each spot in the embryo. We can see, for example, that it's that cardiomyocytes and endothelial cells in the heart both express FGF9, but only cardiomyocytes express the receptors for FGF9. So you can imagine doing cool ligand receptor analysis and things like that with these data. What I'm showing you here is what's called a Kriging plot. And this is an example of the kind of thing that we need to do better at with single cell analysis software. This is sort of off-the-shelf spatial statistics code that we ran, but there's a bunch of things about this that could be made a lot better, and that's one of the things that we're going to be working on in Monocle. So we're going to be trying to enhance Monocle to support SkySpace and other types of spatially resolved single cell analysis, use it as a platform for building new tools for spatial transcriptomics down the line. So I just want to conclude by thanking the development team for Monocle. The lead developer now is a man named Brent Ewing, who actually worked on FRAP and FRED, so he's got lots of experience in bioinformatics. Monocle was originally written by me, and then by Jiaji Chu, and then again by Hannah Pleiner. Hannah is still with us as a kind of a consultant on the development team, and Jiaji keeps an eye out for us and from his postdoc at UCSF. I want to thank CZI in particular for support for this project, as well as some other funders, the Brotman Beatty Institute and the Paul G. Allen Frontiers Group. Monocle's been supported over the years by different ways, of course, the NIH, but it's really hard, as many of you know, to get funding for this kind of software development. With NIH, you kind of have to sneak it into other grants. So it's really incredibly nice and good that CZI has a mechanism for supporting open software like this. I really appreciate it. Thanks very much, and if there's any time for questions, I'd be happy to take them.
Cole, that zebrafish example you gave, it sounds like it would also be really good for some of the organoid cultures our group's working on. Yes. So we have a couple of organoid collaborators and are starting to use that for organoid systems. I think I would really be excited to do experiments where you actually are labeling the cells by which individual organoid they came from, and we think that will be possible. At least for some systems and over a certain range of their development. Like when they're really early and tiny, it's going to be tough. And when they're really huge, it'll probably work, but you might run into other challenges just running the system that long. So I think there's going to be a kind of a window where it's super helpful.
Just wondering what the resolution of the SkySpace is.
So right now, because we used a microarray printer to make these from like 1992, I think, or something, it's around 200 microns. But we think that the resolution of the assay is limited by two things. One, the geometry of the grid, which if we had access to more modern printing architecture could be way higher. I think we could probably get it down to less than 50 microns, maybe even smaller than that. So right now it's around 200. The other limitation, and this is harder to evaluate, is the diffusion of the oligos off of the initial slide and onto the tissue. So we can kind of get a sense from that based on looking at, so some of the spots on the grid are actually, we have cyber doped into the spot, so you can see like how big the actual footprint of the cyber is. But of course, DNA molecules are going to diffuse differently than cyber, so it's hard to know exactly. But we think that we can tighten both of those up with better kind of fabrication techniques. the future generation of the array design.
Awesome. And I just wanted to mention one thing. Great work with Monocle, and I really like the idea of making it more modular, because we've already needed to kind of change batch correction methods or other things like that. It'd be great to be able to have the different parts of at least trajectory inference split up and accessible. So thanks.
Hi, thanks for the interesting presentation. I think like all of the parts were very, very cool work. I was wondering about the size space. You mentioned that it would be like such data would be really cool for ligand analysis. Is this something that you're already working on, and like how far are you along with that?
Yeah, we're working on it. You know, I would say we're along. We're not. You know, I think what we're trying to do is a bit, you know, there's been some techniques that have been described, like cell phone DB and things like that, right? Which are cool. I think we're focused on a couple of aspects. One is that sort of takes advantage of the unique features of sky space. So sky space has a couple advantages over, and there are some disadvantages. A couple of advantages over other spatial transcriptomic technologies. So it's really fast to do the experiment. So unlike something like seqfish or merfish, where the imaging itself just takes a long time, you can do a lot of tissue. So you can look at really big experiments. You could do, you know, serial sections or things like that. You can collect a lot of data, lots of data on all genes. Spatial resolution is much lower. So we will not get, like you get in merfish or seqfish, we will not know that two cells are touching one another. And it's like a sampling of cells from the tissue. It's not every cell. So those are huge differences in terms of what the data looks like. And then unlike Visium, it's single cell. It's not like spots that have contributions from a bunch of different cells. So I think there's some cool things that enables in an algorithm, but it also limits the algorithm in some ways. So we'll see. You know, I don't have any results to report to you other than just I can report hope and optimism that we'll have something cool. But if you're working on it, don't feel threatened. You should do your thing, because we're not going to get you.
I'm not working on it, but Robin Browijs is working on or recently published NicheNet, which also involves ligand target predictions. I found it interesting that the talks so far in this session, all of them mentioned in some form or some form of another file formats conversions, and found it funny that this was also one of your points. It makes me think that maybe at some point we should just all get together and like try to decide on things, because it seems that like so many people are doing a lot of work in this area, or doing a lot of work in this area, and sometimes in parallel. So I don't know.
Yeah, it's a bit of a thankless task, though. I mean, I think the reason that it's still kind of an unsolved problem is because it's really hard, and like, you know, and everybody realizes it's super necessary, but it's not the most fun thing.
No. Also, everybody has their own specializations, right? I mean, I might be interested in trajectory inference, somebody else in spatial. So whenever you're writing converters, these converters are also going to be focused on a particular application more often than not. We should introduce a new file format that can act as an in-between for all the different files.
I think there was a meeting, yes, regarding file formats between Alex, who is initial developer of Scanpy, and someone from Seurat. I don't know. I mean, but I think
Seurat disk. Were they talking about Seurat disk, which can convert between Seurat and
I'm not sure I know the details. I just know that like. Yeah.
See, so a lot of people are doing this very much in parallel. Anyway.
Hi, my name's Mary, and I'm here to talk to you guys about UCSC Xena. As I said in my brief 1-minute pitch, Xena is a web-based browser for visualizing and analyzing cancer genomics data. So again, we're looking at gene expression, which I know a lot of people here have been talking about, also mutation, copy number, that sort of thing. Our strengths is that we provide easy access to public cancer genomics resources like the TCGA and other resources out there. We also have a mechanism to securely and privately view your own data or data from the literature. So we support both of those data resources. We visualize and integrate multiple different types of omics data. So that's really, a lot of people come to us for that, because you can view mutation, copy number, gene expression, DNA methylation, proteomics data. You can really integrate all of this data together with us, as well as a number of different phenotype parameters, such as age or the size of the tumor, that sort of thing. We're really built for scale and performance. So like I said, we support single cell analysis, and then we also support large public bulk sequencing data sets. And then we have a really wide variety of visualization analyses and statistics. So some example questions of things you can ask with Xena are things like, is this gene differentially expressed in tumor versus normal? Or is the overexpression of this gene associated with lower or higher survival? Or what is driving the increased gene expression for this particular gene? You know, is it looking like there's something, you know, a mutation, or is it, you know, a copy number alteration, that sort of thing. So for our CZI grant, our whole goal with our CZI grant was to better support our users. Thank you. We're very excited to have done some of this work and are continuing to do more of it. We are better supporting our users in 3 aims. The first is improving the usability of 2 of our key features, and that's going to be my first part of this talk. And then we also have 2 other aims, which is to standardize and modularize our training materials. And we've already started doing this, but I also got a lot of really great ideas at this conference, so I'm really excited that I could be here and could present as well. And then we want to build community through a blog of Xena-focused publications. And this will be really useful not only for users to kind of see what other people have done with Xena, but also give them a place to, you know, comment and like, and you know, possibly even find collaborations. So I'll be talking about how we improved usability, which we did this mostly through user experience design. I'm sure a lot of people here already know about this, but you know, as far as when you ask, well, what makes a tool easy to use? There's kind of 2 categories. First is just some general best practices, right? Making your website accessible to people who might be colorblind, using consistent buttons so that people recognize, oh yeah, that's a button on my website, perfect. But a lot of it also depends on the particular tool, the users, what their motivation is, what their skill set is, and also the environment in which they are using the tool. So user experience design really tries to take this into account. It is designed based on an explicit understanding of users' tasks in the environments and is refined by user-centered evaluation. So we use this to redesign 2 of our key features. And we started with, oh, sorry, there it goes. We started with reviewing user feedback. So we looked at from 3 different places. First, just from watching users in workshops. You're giving a workshop, people are working on their own, and you can often notice places that people are getting stuck, places where they're confused, that sort of thing. We also looked at mailing list questions. So even if somebody asks a question and I have a help page up for it, and so I can just point them to that, the fact that they're asking the question shows that it's not necessarily intuitive from the interface itself how to get to you know, the graph or the analysis that they're looking to do. And then we also solicited direct feedback from power users, including people from our team. So basically, you know, these are people who are using Xena very frequently, and just asking them and talking to them, seeing what little tweaks or things that we can do, or even bigger features that would really help push their analysis forward. And we took all of this feedback and we used it to brainstorm some new designs. And then we took those new designs and we started building prototypes of them. So we built prototypes in two different technologies. The first one is Balsamic, and you can see a sort of a screenshot of one of our prototypes here. And you can see it kind of has this kind of like blocky look to it. And so this is really helpful for getting really frank feedback from users because it doesn't feel like a finished product. Users are often much more, feel more like they have permission to open up with like, oh, I just hate the entire thing, or, you know, whatever it is that they are thinking. We also built prototypes with InVision, and you can see here another screenshot. It's much more polished. It actually feels much more like the real website. And this is really good for getting feedback on user workflows. So, you know, we think that we've put the action very close to the, you know, the done or the close, but people are still struggling to find it. And so we need to move those two things closer together. So after we had built these prototypes, we decided to do some user interviews, and I put interviews in quotes. That's kind of what it's called in the user experience design, but it's not really an interview in the traditional sense of that. So we recruited users for 1-hour interviews, which is basically just, we give them a task, which is a common task that people will be doing with their website. And then we just watch them use the new designs. These designs are actually, either can be on the web, which they're still just static pages, but we've made little clickable elements. You can also do it on PDFs, and we did both of those. And then as they're going, we just watch, oh, they're getting stuck there. They're confused on this piece. That part worked really well. We also ask them to think out loud. You know, so, you know, when they're looking for something, they're like, oh, where is that particular thing? So kind of gives a little insight into what's going on in their heads there. And then we use the observations from these interviews to iteratively refine the designs. And I just want to give sort of a little, one very small vignette of the user interviews. I'm laughing because we got so much insight from these things, and this is really just one of the, you know, very 1% of what we got, but it really shows the power of doing these. So at one point in the process, we had kind of two prototypes that we were looking at. One is what we call the wizard prototype. So you have a certain amount of data on the screen, and then they want to create another graph off of it, another type of analysis. And we just ask them, okay, well, what do you want to do? Do you want this, this, or this? Excellent. Do you want it like this or like this? Excellent. And we just went through the questions and then gave them the graph. There was a lot of clicking, but we thought that this might be something that people would like. And then we also did this, what we call the show all prototype. So again, you have a certain amount of data on the screen. Well, in Xena, there's not an infinite number of graphs that you can create from that data. And so what we said was, well, why don't we just show them all of the graphs that we are able to make? And then because we have, you know, we have data on this, is to just show them the graphs that they're most likely to be interested in at the top, and then just put the rest of the graphs there at the bottom, and they can browse through them, kind of get a general feel for what the data looks like, how it distributions and that sort of thing. And so we showed both of these prototypes to people, and while people did like both of them, what was surprising to us is that actually people preferred the wizard prototype, despite the fact that it had a lot more clicking than the show all prototype. And what the feedback showed was that users really felt empowered to make the graphs that they wanted. You can see a quote here at the bottom from one of our users. You know, what do you want to do? There's that sense that you have control over what is being made. I like that you go through the process yourself, right? So people really felt like I am controlling what's going on rather than just a bunch of graphs coming at me. And then completely, we had no idea that this was going to be a piece of feedback, but people said that that the wizard prototype could make the most types of graphs compared to all the other prototypes, which is actually a complete lie. All of the prototypes can make all of the same types of graphs, but by essentially listing them, by asking people about them, people felt the wizard prototype was more fully featured. So yeah, so this is just one of the many vignettes that we got from doing these interviews. So our next steps is we have our finished designs. We're finishing implementation. We're quite close, actually. We're very excited about it. We'll be putting it on dev here pretty soon. Then we're going to do user acceptance testing. So basically just have users run through the actual program as it's going, the actual features, just verifying, did we actually do what we set out to do, make any last tweaks that might need to be made, and then release to production. So now I'm going to change gears completely. This is not something that was part of our CZI grant, but I thought might be interesting for people here, which is to talk about how we are integrating ourselves into cloud compute environments, leveraging Jupyter notebooks. So our motivation is that everything is moving to the cloud, as we all know. But there's actually a lot of different cloud environments for bioinformatics and bioinformaticians. Some examples are things like Terra or Gen 3, ISB Cloud, and they all have their own different types of user security, logins, all that type of thing. And so we were looking at this and we wanted to be able to support users who want to run their analyses in all of these different spaces, not just one, and to visualize that data in the web-based browser that we have. So going back, if you remember, I said that we provide visualization analyses on both public cancer genomics resources and then also on private data. And I just kind of glossed over how we do that. I'm going to give you a peek under the hood now. So our architecture actually has two completely distinct components. We have the Xena browser here, which just runs in a regular web browser. And then that Xena browser talks to any number of different Xena hubs. And those hubs can be located in lots of different places. You can see here we have some, this one is running on the person's laptop, so they've done their own analysis. They're now looking at data from it. There's also a number of public hubs. I realize I should have put a little cloud about this. A lot of these are hosted in the cloud themselves. This has data like the TCGA data that I was talking about before. So now we wanted to support, the cloud compute environment, lots of different types of them. And sort of a first pass was like, well, great, we'll just put our hub up in there too, and then we'll be able to support it. But then as we started looking at this with all the different security models, it quickly became clear as like, how are we going to get that data to the Xena browser on the person's laptop so that they can visualize it and not do it just for one, but do it for many of them. And so what we decided to do was to leverage Jupyter notebooks. So most cloud computing spaces support Jupyter notebooks, so we don't need to make that functionality available. And then also it is building off of knowledge that many bioinformaticians already have. They already are familiar with Jupyter notebooks, and many of them are using them. So I'll just take you through our prototype that we've built of this end-to-end system here. First, this is the steps that a user could take. So somebody starts up a Jupyter notebook, they run their single cell analysis, any number of the wonderful tools that have been talked about already and will continue to be talked about. At the end of the analysis, they can initiate a Xena hub in the same place, load the analysis results into that hub, and then visualize that data. So now I'll show you what sort of is happening behind the hood here. So we have the web browser, and we have a Jupyter front end and a Xena browser running in the same web browser. So these are just two tabs in the same place. The Jupyter front end is communicating with the notebook server via the Jupyter Communication API. And so they've run their analysis here. And now they spin up a Xena hub. They now load that data into that Xena hub. So now the Jupyter front end can read data from the Xena hub through these connections. And then we have the Jupyter front end and the Xena browser actually talk to each other via cross-tab communication, post message here. And so now the Xena browser can now talk to this Xena hub going through the security that is already being provided by Jupyter. So our next steps are to harden integration. So right now, you really have to do everything in a particular order. And if you don't, then things don't work quite right. So make it so that we help people out a little bit better there. Adding connections to single cell analysis tools. So this harkens back to the conversation right before I started about being able to translate between different formats, and then also adding some help and tutorials to support our users. With that, I'd like to thank you. This is our team. Thank CZI and NCI, and yeah, I'll take any questions now.
That was great. Thanks, Mary. And so at the beginning of your talk, great example of the power of the user experience and identifying key elements that make it easier for people to use tools in that. We do it a lot at CZI for some of the text development that we're doing. So nice example. And just in terms of how you set it up was nice in the interview, walking it through. Have you applied it anywhere else, or have you had people interested in sort of using your approach?
So using experience design in as part of the design process, coming up with tools. Yeah, I mean, we use it. We've done it for a bunch of other different features that we've had, including like our data loading wizard, and then what we call kind of our like onboarding when you first come to Xena to be able to make your very first visualizations and that sort of thing. So we've done UX design for those as well. And then, yeah, we actually, I'm from UC Santa Cruz, and so the Genomics Institute, there's various people here who are using it. And then if I'm allowed to, actually, I'm going to give a quick plug for a workshop that I'm going to be doing that's going to be happening. It's at PSB, which is normally held in Hawaii, but is online this year, unfortunately. But it's free to register. And I'll be giving a workshop with a bunch of other UX designers as well. So, and that's happening, I don't have the exact, it's very early in January, that first week in January after the New Year's.
Great, thanks. And Robert, you have a question?
Yeah, so I'm very curious because unfortunately I hadn't heard about Xena before, but now after seeing your presentation, I'm very eager to give it a spin. And especially so since I didn't really see many screenshots or videos of the interface itself. So now I'm even more curious. Maybe I should attend the workshop in not Hawaii to find out more about that. Could you, sorry?
Oh, I was going to say the one in Hawaii will be actually more similar to this talk, and that it won't be very much about the visualizations and more about the process. So, but we have a number of tutorials and workshops, just stuff that you can see on our website and our help material if you want to get a feel for it.
Cool. Could you give us, or could you give me some insight in how Xena was started as a project, like with what motivations was it created? Because I hear that you're listening a lot to user feedback, but I assume that this project was created because somebody locally had a need to do this type of visualizations.
Right. So we started out, and I'll just say we started out with bulk sequencing data, and then we've only just recently put our toe into the single cell world, and we really got started because people were, so originally the tool that predates this one is the UCSC Cancer Browser, which visualized just data from TCGA only. And people kept on coming to us and saying, oh, this is so great. Can I give you my data to visualize? By the way, it's all private. We're thinking, oh my gosh, how are we going to support all these people and then keep it all private and the security and the logins and the da-da-da-da-da. And so we went back to the drawing board and we're like, how can we make it so that people can visualize their own data easily alongside that public data? And so that's where we came up with our design. And I didn't really talk much about the hubs, but the hubs are super, super easy to install. We've made it, it's like a point and click GUI interface. There's no command line or anything. We support Mac, Windows, and Linux. And we're really trying to put it into the hands of people who may not have very strong command line skills.
It sounds like a lot of engineering went into making this project, and without it being, and even more so, you're still doing all of these user experience studies. So that's really surprising in a data science context, but really cool.
Definitely. And I was just going to add really quickly that I'd like to encourage everybody to do at least a little user experience design in their work. Even if you just talk to one person, you'd be surprised what you hear from people. So yeah, excellent.
So I will talk a little bit about the dynverse project, which is more specialized towards specifically running trajectory inference analyses and everything that surrounds this. I mean, we've heard about lots of different toolboxes in these sessions, and they're all very fantastic in their unique special way. And the unique special way that we're trying to tackle this is with a specific focus on analyzing dynamic processes within single-cell data sets. So a little bit of background on what trajectory inference is. Basically, you start off with your single-cell omics data set. And when you run trajectory inference, the type of output that you get is, well, it's a prediction by a computational tool. And the type of output that you get is, a topology where your cells are being mapped onto. And a graph view is one of the ways that you can visualize a trajectory, but one of the more useful ways to really perform downstream analyses is with a heat map where the trajectory is shown alongside of it. Because now you can really see which genes are changing in expression along the trajectory in a bird's eye view. Of course, it's also very useful to visualize these in a dimensionality heat map, perhaps by performing some kind of trajectory differential expression. This is something that we're looking at as well, together with Koen van den Bergen with his trait-seek approach, where we aim to look for genes that are differentially expressed along a branch or between different branches in a nice pattern. And what this allows you to do is to create a smaller visualization of a heat map where you see all of the genes that are specifically differentially expressed for one of the branches. And then finally, with our toolkit, you can then relabel all of your milestones in order to be able to gain or to map your current biological knowledge from your data set onto it, or extract new findings from it using the heat map view. So trajectory inference started in 2014, way back in 2014, with methods such as Wanderlust and Monocle, and these were really instrumental in defining what you could or couldn't do with trajectory inference. And since then, the field has really exploded, and it's also very diverse. You can see that from the types of outputs that the methods produce. So each of the methods will produce one or more of these different topologies, which can range from linear, bifurcating, a tree, a cycle, or even disconnected. And we saw that they were we tried to bring that into light in a review paper, where we described each of the trajectory inference methods as in kind of like a subway plot, subway map, I mean, where we split apart the different algorithmic components that each of the methods subsists of. And by now, of course, there are more than 70 methods that have been developed for performing trajectory inference, and trying to create a subway view of how each of these methods work is near impossible, I believe, or you would have to start doing some kind of a large view pathway analysis type of visualization. And my colleagues will groan when I show the following plots, because they hate when I talk about this. But unfortunately, the field, especially at the start, and it still kind of is a little bit of a wild west, where lots of people seem to be publishing peer-reviewed or preprint articles introducing a new trajectory inference method without performing some kind of a quantitative analysis or benchmark of their tool. And I can totally understand that, especially at the start, because single cell data sets were especially scarce, so it was hard to perform benchmarks. It was hard to compare against other tools because the different types of output are so varied. But we wanted to tackle this problem a little bit, and that's where the dynverse project really got started. Together with Wouter Saans and Helena Todorov and Ivan Sais, we performed a large-scale benchmark where we collected 110 real single-cell data sets for which there was a ground truth known, and about 200 synthetic data sets which we simulated with various simulators. And we evaluated 45 different trajectory inference methods with 4 different metrics. And this is, again, an overview of all of the results that we got in that benchmark, split into the 4 different categories: accuracy, scalability. So those are our metrics. And we found it important to not only analyze the accuracy of each of these tools, but also how scalable are they and how stable are they when you run them multiple times. And interestingly, this has a little bit of a connection to the Xena presentation shown before. We also evaluated how usable each of these tools are. So this is the top of the heat map, showing only 15 components. So we looked at how usable each of these components are, methods are, with regards to their software and documentation that they provided, and found that some of them are actually very well developed in that regard. What we found in terms of benchmarking was that there was still a large variability in the scores, in the overall scores that each of the methods obtained on the individual data sets. And this could be largely explained by the type of underlying process that was inside of the data set. So whether it was a linear process or bifurcating or something more complex. So in that regard, we made a set of guidelines for which trajectory or which combination of trajectory inference methods you should use. But by now I feel that these guidelines are a little bit outdated and should be updated soon. But this requires, of course, running another large-scale benchmark. So now I will go through a little bit of the developments that we went through over the past year. First off, we introduced, or we published a preprint on bioRxiv containing the DynGen simulator, which is a simulator for trying to benchmark single-cell omics tools in a broad sense. How this works is it uses a gene regulatory network, which is constructed and by sampling real networks and doing some specific modeling of what gene regulatory networks in some dynamic processes might look like. And what the simulator will do is that, like, at any given point in time, the state of a cell is described by the molecules it consists of, and depending on the molecule numbers, any of the different biomolecular processes will be executed throughout the simulation, such as transcription, splicing, etc., etc. And so from the simulation, we can extract ground truth information that allows us to benchmark single-cell omics tools. For example, you can simply extract a cellular state and what point of the cellular state a cell is at any point in the simulation. That allows us to benchmark trajectory inference methods, but also trajectory alignment methods. We also can look at the amount of times that a particular reaction is occurring within a specific cell at any point in time, which allows us, in this case, to benchmark RNA velocity methods, such as Azimuth. And also we can take a look at the amount of regulation that is occurring within a cell by another transcription factor at any given point in time. And this allows us to benchmark cell-specific network inference, which I think is a really cool topic. So within the DinGen paper, we did do a minor benchmark on all these three types of single-cell omics tools. But I have to say that the benchmarks that we performed were really simplistic, in the sense that we only looked at the accuracy of a tool for these kinds of data sets, and this should be supplemented with other types of benchmarking, looking at the stability, the scalability, and also using real data sets whenever possible to also perform some kind of quantitative benchmark. Another thing that came out of the benchmarking story was that at some point we had 45, now 55 different trajectory inference methods, and we've wrapped all of these inside a common interface where your inputs are well defined, so you can have the expression data, but also varying optional prior inputs, as well as the output that is standardized. All of these methods were wrapped in individual Docker containers. These were all wrapped in Docker containers, which are either Python-based, R-based, and also R and Python-based. So for this, I created a container called RandPy, or RandPy, which contains both R and Python versions. And you can choose whichever Python or R version that you want. So if you want R 4.0 and Python 3.7, that's totally a thing. This was very useful in being able to adhere to the nitpicky requirements of each of the different trajectory inference tools. So what we realize is having all of these trajectory inference methods wrapped inside of a common interface, we could start up develop a toolkit that leverages all of these trajectory inference methods and be able to perform downstream analysis for trajectories. So one of the things that you can do is to determine the directionality of your trajectory, either manually using prior knowledge from gene expression, or now we're also looking at RNA velocity to determine the directionality. You can run various sorts of differential expression tools that allow you to detect particular genes of interest. And you can also visualize the trajectory in a myriad of ways, either as a heat map or as a stream plot or something else. You can compare different trajectory outputs on a common dimensionality reduction, which is not a trivial task to do, but we managed to include it. And some of the frequently asked questions that we got for Dino was actually simply, how can I run Dino on my H5AD file? So we're looking at adding support for importing H5AD and other objects. I mean, we've also had requests for Loom and Seurat and single-cell experiments, but there I think the boundary or the thresholds to be able to manually import that into Dino is a little bit less high in comparison to H5AD when you're in an R ecosystem. How can I customize my plots? I mean, we have many different types of visualization, but the customization had to be done through parameters. So sometimes, not all of the parameters were added if you wanted to change the font size of particular labels or something. So now we provide a more modular, customizable interface that uses ggplot2 to be able to add layers to your plots. Also, can I use RNA velocity to determine the directionality? This is something that we're working towards. Won't be showing today, but hopefully soon. And can more of these cool trajectory inference methods be added? Because they're not in the ecosystem right now. So also there, we're adding functionality for more and updated versions of particular trajectory inference methods. So I'd like to highlight a little bit the relation between Dino and OnData, because especially the question, how do I run Dino with my OnData file, kept me awake a little bit, so to say. And I think the underlying question here is actually, how can I use H5AD files in R? Because I think that's the major roadblock here. For now, I think you had three major ways to tackle this problem. Either you can directly interface with the H5 files using HDF5R in R. It's fairly easy to do. However, there's a lot of manual work involved because OnData is doing a lot of things under the hood to convert your Python objects to something that is storable in H5AD. So trying to get that back out is a major hassle. Also, if you change something to your H5AD file and then save it back as a file, and then try to reload it in OnData, this might not work because of incompatibilities in the HDF5R library, especially some bugs that are still in there, caused major headaches. And if you're doing this a lot of times, then you have copy-pasted code everywhere, and it's an ugly thing. You can also use reticulate to directly access the Python interface from R and be able to work with that. However, well, the command is also very easy, and it allows you to do a lot of things. However, there's still quite a bit of manual work involved because in order to convert some of the custom classes that OnData defines, because of course reticulate can't know how to convert these classes, and so that's still requires a bit of code, which is of course, if you have multiple data sets, multiple scripts, gets copy pasted all over the place, and it turns into an ugly story. And lastly, you can also convert your H5AD to Seurat or single cell experiment or some other kind of data structure which does exist in R. And I think this is mostly the best way to go, especially because when you do this, you automatically get a lot of R pipelines for free that already exist that allows you to run on Seurat or single cell experiments or etc. However, and this is the big however for me, if you want to stick to H5AD, for example, you have a complete pipeline which is working with H5AD files, and in a single step you need something to run in R. In that case, I want something lightweight that doesn't convert from H5AD to something else in order to be able to convert it back afterwards. And so for that, I made a small package, or turning into a larger package, that uses reticulate to access the OnData interface. But the package, which is maybe confusing me, also called OnData, converts the whole API into something that feels very natural in an OnData environment. I have a few examples on the GitHub. So by installing the package, you can create an OnData object, which is very, very similar to what you would do in Python. You can access all of the objects that are inside your file. You can read a file you can read an OnData object from an H5AD file. You can create views. And something that I found very surprising was actually when you use reticulate to import Scanpy. So you're still in R. You can pass along your OnData objects from the R interface. It will do lots of stuff in Python, and then at the end, your AD object is still an R object, so you can still do all of your R stuff with it afterwards. And I was very surprised when this works so nicely without. Well, this I actually got almost for free based on how reticulate worked. So it was very cool to find out. So OnData, it's on CRAN, but don't use that version. I'm porting the Python unit tests to R to detect discrepancies. And then I won't talk about the customizable Dino plots, which, yeah, in order to save time. Thanks for your attention.
Hi, great talk, Robert. I was just
Thanks.
curious as to what sort of issues you were having with Seurat. I would think that because both are in both the dynverse and Seurat are in R, it'd be very easy to use Seurat, and we are trying to make Seurat as easily extensible as possible. So anything that you notice that's hard to do in Seurat, I'd be happy to fix.
It's very sweet of you to ask this question. To be honest, I don't have any problems converting Seurat to, using Seurat objects in order to run trajectory inference. But you do need to manually take, as it stands right now, you do need to manually take the expression data and cell information from your Seurat object and put that into the dyno objects. Mm-hmm. Because there's no function that allows you to just convert a Seurat object to a dyno object. And so I think some of the users kind of expected to be able to take a Seurat object and just plug it into to dyno without having to manually access the expression object. So I think all I need to do there is just provide a function that says read from Seurat or read from single cell experiments. But I kind of focused on the on-data part first because that was the hardest part for me, because I needed to do a lot of work before that. And I think converting from Seurat is actually pretty darn easy in this specific case.
Okay. Well, if there's any help that you need, feel free to reach out.
Thanks. That's very kind to offer.